Imagine a world where the boundaries of biology are rewritten, where the once rigid rules of cellular development are challenged and redefined. This isn't just a flight of fancy, it's the reality brought forth by the groundbreaking work of biologist John Gurdon in the early 1960s. In an era dominated by the belief that cells, once specialized, could never revert to a more primitive, undifferentiated state, Gurdon proposed something radical. The scientific consensus of the time firmly held that the fate of cells was sealed as they matured. A nerve cell, for example, was destined to remain a nerve cell forever. Enter John Gurdon, armed with little more than a microscope and a revolutionary idea. He embarked on an audacious experiment using the humble frog as his subject. Gurdon took the nucleus from a mature intestinal cell of a tadpole and implanted it into an enucleated frog egg, a cell stripped of its own nucleus. What followed would not only defy the scientific dogma of the day, but also lay the foundation for modern regenerative medicine. The egg, now housing the nucleus from a fully specialized cell, began to develop normally. It didn't just grow, it blossomed into a fully functional tadpole. This was not just any tadpole, but a living proof that the mature cell's fate was not as fixed as once thought. The implications were profound. Gurdon's work demonstrated that the genetic material within a specialized cell could be reprogrammed, reverting it back to a state from which it could develop into any type of cell. This revelation opened the floodgates to a new understanding of cellular development. Decades later, this experiment would pave the way for cloning and advancements in stem cell research. Scientists, inspired by Gurdon's work, began exploring the possibilities of reprogramming adult cells to form new tissues or even new organs. For his pioneering contributions, John Gurdon was awarded the Nobel Prize in Physiology or Medicine in 2012, sharing the honor with Shinya Yamanaka, who expanded on Gurdon's findings to create induced pluripotent stem cells. Together, their work has not only reshaped our understanding of biology but also holds the promise of revolutionary treatments for diseases previously deemed incurable.